Hello and welcome to this Microbiology Bio 203 video lecture from Chapter 8 of your OpenStax book, Microbial Metabolism. I'm Mr. Kennedy and I'll be your guide as we explore this topic together. Learning outcomes for Chapter 8 are listed below and include defining metabolism, anabolism, and catabolism, defining autotrophs and heterotrophs, comparing and contrasting oxidation and reduction, and being able to describe high-energy electron carriers. We begin today by looking at metabolism. Metabolism is the sum total of all chemical reactions in the cell. It's actually a combination of two other reaction types, catabolism and anabolism. Catabolism is a series of reactions that is thought to break large molecules into smaller ones. It often releases energy, while anabolism is the exact opposite. Anabolism are reactions that take small molecules and assemble them into larger ones. They often involve an energy input. ATP, or adenosine triphosphate, often shuttles energy between the two different types of reactions. Seen below, we have a series of disks undergoing anabolic and catabolic reactions. When the disks are separate and energy is added, they can be linked one to the next through an anabolic reaction. Once linked, they can undergo catabolic reactions that separate them and release energy. Autotrophs and heterotrophs. Autotrophs and heterotrophs can be defined according to carbon source or to energy source. When defining them in accordance to carbon source, we look at carbon dioxide and organic compounds. Organisms that use carbon dioxide are plants, cyanobacteria, and the alga, and they're considered autotrophs. If they use organic compounds, they're considered a heterotroph. Autotrophs and heterotrophs can also be sorted in accordance to their energy source. When looking at their energy source, if the energy comes from the sun, they are a phototroph. If the energy comes from chemical bonds, they are a chemotroph. Organotrophs are organisms such as humans, fungi, and many bacteria, while lithotropes are other microbes. This table will provide you with a summary of organisms by nutritional type and also indicate their energy and carbon source. Before moving on, pause the video and take a moment to familiarize yourself with some examples of organisms in each nutritional category. Redox reactions. The word redox is actually a blended word of two longer words, reduction and oxidation. Reduction and oxidation reactions are metabolic reactions that often involve the transfer of electrons. Oxidation reactions involve the loss of electrons, while reduction reactions involve the gain of electrons. There are several acronyms that can be used to help remind you of this fact, that oxidation is loss of electrons and reduction is gain of electrons. Acronyms like oil rig or even Leo-Ger can accomplish that goal. Redox reactions are always coupled reactions, a reaction where one atom or molecule loses electrons and the other gains. Oxidation reactions often release energy. Think of burning things. Energy is transformed into heat and light. Live organisms release energy in a stepwise manner through electron transport chains to be safe. How do cells get energy out of organic molecules? Consider the equation on this slide. Glucose plus oxygen yields carbon dioxide, water, and ATP. If you take a moment to think back when you've seen this reaction before, you might remember it from a biology class dating as far back as high school. This is the simplistic reaction series of cellular respiration. In this reaction sequence, energy is released when the chemical bonds are broken in glucose. Glucose is oxidized to carbon dioxide and water. ADP harnesses that energy to form ATP. If you burn sugar, all the energy will be released as heat and light. There is so much energy released simultaneously that it's actually biologically dangerous. Therefore, in living systems, the standard way of releasing energy from sugar involves doing it gradually, creating almost a ladder of energy. The energy will then be stored in ATP and harnessed safely. ATP, or adenosine triphosphate, is the energy currency in the cell. ATP shuttles energy everywhere it is needed. To make ATP, an additional phosphate must be tacked on to ADP. ATP has 3 phosphates, ADP only has 2. When we add a phosphate to ADP, energy must be input. This energy can come from a gradient, either produced chemically or through light. This energy can also come directly from enzymes, from our food. In either case, energy has to be input. Adding a phosphate to ADP is called phosphorylation. That energy will be carried in ATP until needed. Once a reaction is reached that needs energy, the third or terminal phosphate will be removed from ATP and energy will be made available to do cellular work. This is called dephosphorylation. ATP and ADP will continue to oscillate from one form to the other indefinitely. Exergonic and endergonic reactions. Exergonic and endergonic reactions are coupled reactions. Reactions in which reactants have less free energy than products are called endergonic. They will not proceed without a net energy input. Reactions in which the reactants have greater free energy than the products are exergonic, and they end with a net release of free energy. Storing energy. Energy can be stored in one of two strategies, short-term or long-term. Short-term energy storage involves ATP and similar compounds like GTP. Short-term energy can also involve high-energy electron carriers such as NADH, FADH, and NADPH. These electron carriers can exist in one of two different forms depending on whether they are oxidized or reduced, as seen here. Long-term storage of high-energy compounds involves biomolecules like carbohydrates and fats. Sugars and polysaccharides, such as starch and glycogen, are good high-energy storage compounds for quick energy release. Fats have higher energy content than carbohydrates, as much as 9 times on a gram per gram basis, but their energy release is slower. Enzymes. Enzymes are proteins that are usually globular with complex three-dimensional structures. Enzymes behave as chemical catalysts, decreasing the activation energy needed to get a reaction to begin. The reactants of enzymes are called substrates. Substrates bind to enzymes at their active site. Many enzymes also have an allosteric site. Think of the allosteric site as an on-off switch for an enzyme. It allows the modulation and control of enzyme activity. Here we see how enzymes reduce the amount of energy needed to begin a chemical reaction. The graph on the left shows a steep peak separating the substrate from product as we move from left to right. That peak is the activation energy that must be satisfied before the reaction can continue. On the right, we have the same reaction sequence, but an enzyme has been used and the peak separating the substrate from product has been reduced. Enzymes make a reaction run much faster than it would on its own without being changed by the reaction. Here again, we see how an enzyme can lower the activation energy of a reaction. By following the green and red dashed lines, you'll notice the difference when an enzyme is used and when one is not. Exactly how an enzyme reduces the activation energy can be understood through the induced fit model of enzyme action. Here we see the substrate connecting to an enzyme at its active site. At one point in time, it was thought that this union was so specific that it was described as a lock and key fit. As we've learned more about enzymes, we've learned that it is not a lock and key fit, but instead a strained or induced fit. The substrate fits into the active site, but does not do so perfectly. And as a result of this imperfect fit, a strain is placed on the bonds of the substrate, which causes it to come apart or to react with less effort or energy input. Enzymes are classified based on the reactions they catalyze. Here's a list of a few examples. Oxidoreductase is an oxidation reduction reaction enzyme. Transferase moves functional groups around from molecule to molecule or within the same molecule. Hydrolase is a hydrolysis enzyme, and so on. Native structure and denaturization. In order to function, enzymes require their 3D native structure to be intact. If they are exposed to extremes in temperature or pH, they may unfold even though the peptide bonds remain intact. Once they unfold and lose their three-dimensional structure, they are considered denatured and will no longer catalyze a reaction. Complex enzymes. Enzymes known as holoenzymes are made up of a protein subunit of the enzyme called the apoenzyme and a cofactor or coenzyme. A cofactor would be an inorganic add-on to the apoenzyme. A coenzyme would be an organic add-on to the apoenzyme. Putting the non-protein part and the protein part together makes a complex enzyme that again is referred to as a holoenzyme. Factors affecting enzyme activity include temperature, pH, substrate concentration, and inhibitors. Here we see a graph illustrating the effect temperature has on enzyme activity. Notice the warmer that it gets, the greater the reaction rate for the enzyme. However, there is a limit to this enzyme's ability to perform at high temperatures. If you look closely at the graph, you will see it at about 40 degrees Celsius. The graph peaks and then falls off dramatically. This is an indication that beyond 40 degrees Celsius, the native structure of the enzyme is lost and it will denature, losing its function. This is a graph illustrating how pH affects enzyme action. Note, again, that there are peaks and valleys to this graph. Every enzyme has a specific pH that it prefers. In most cases, that pH is centered around neutral. There are, however, some enzymes that work best in an acidic environment while others work best in an alkaline environment. In any case, notice what a deviation from normal or optimum pH does to the reaction rate catalyzed by that enzyme. This is a graph of how substrate concentration affects enzyme activity. Notice that as the substrate concentration increases, so too does the action of the enzyme. However, there is a limit beyond which the reaction rate will no longer increase. This limit, or flattening out of the curve as it's sometimes referred to, is the saturation point for the enzyme. At this point, no matter what you do with the addition of substrate, the enzyme will not go any faster. Enzyme inhibitors. Enzymes and their reaction rate can be affected by one of two different types of inhibitors: competitive inhibitors and non-competitive inhibitors. Competitive inhibitors compete for the active site with the substrate. The rule of whoever gets there first plays out in a very real way in this type of control or inhibition. If the competitive inhibitor arrives at the active site before the substrate, the enzyme will not catalyze the reaction. Non-competitive inhibition involves the utilization of the allosteric site or regulatory site of an enzyme. If a non-competitive inhibitor binds to the allosteric site, it may change the shape of the active site and subsequently prevent binding of the substrate. Feedback inhibition is an excellent way to look at the control exerted by competitive and non-competitive inhibitors. In feedback inhibition, molecules can feed back to bind to the allosteric site or to the active site and stop an enzyme from functioning. There are two types of feedback that occur in enzymes. One is negative feedback, as illustrated here. The other is positive feedback. In positive feedback, the connection to the allosteric site may encourage enzyme activity instead of stopping it. Catabolism of carbohydrates. The catabolism of carbohydrates, or breakdown of carbohydrates, is probably the most familiar metabolic pathway that is covered in a typical biology class. The oldest catabolic pathway of carbohydrates is anaerobic and is simply referred to as fermentation. Cellular respiration is the catabolism of carbohydrates in the presence of oxygen. Both fermentation and cellular respiration produce ATP. However, the quantity of ATP produced varies dramatically one from the next. Both fermentation and cellular respiration start with the same reaction, glycolysis, and in glycolysis, glucose will be cut in half to produce pyruvate. Glycolysis. Simply put, glycolysis is the degradation of glucose without the need for oxygen. The most common glycolytic pathway is the EMP pathway. Others include the interdeuterol pathway in bacteria and the pentose pathway, which is supposedly the most ancient form of glycolysis needed to make nucleotides and amino acids. Without going into too much detail, glycolysis will take glucose and break it down into pyruvate. The glucose will be oxidized. There are two phases of glycolysis: an investment or initiation phase and a payoff phase. No oxygen is required for glycolysis to occur. It occurs in the cytoplasm of a cell, and the one glucose that is split will yield 2 ATP molecules in total, 2 NADH molecules, and 2 pyruvate molecules. Here we see the energy investment phase of glycolysis. The reason it is referred to as the investment phase is that glucose is a stable molecule and must be activated in order for it to be split. Here we see the 6-carbon glucose in the far left corner, activated by dephosphorylating ATP, forming glucose 6-phosphate. Glucose 6-phosphate is isomerized to fructose 6-phosphate, and an additional ATP is dephosphorylated, resulting in the formation of fructose 1,6 biphosphate, which is easily divided into two. In the fifth and final step, we have 2 glyceraldehyde 3-phosphates that are formed. This is the energy payoff phase. Here, electrons are transferred to NAD, reducing it to NADH2. ADP is phosphorylated to ATP, and energy is harvested by adding the phosphate group. Ultimately, the payoff phase produces a net of 4 ATP. However, since the investment phase cost 2 ATP to start, we really only gain 2 ATP from glycolysis. The formation of ATP in the energy payoff phase is done using enzymes, which will oxidize the glucose derivatives directly, so we refer to this as substrate-level phosphorylation. Next, we have a transition reaction. This transition reaction takes pyruvic acid from glycolysis. It will oxidize and decarboxylate it, where it will lose a carboxyl group. That carboxyl group will be released as carbon dioxide, which ultimately becomes the waste gas that we exhale. The remaining molecule reacts to form acetyl-CoA. In eukaryotes, the acetyl-CoA will then enter the mitochondria. In prokaryotes, the acetyl-CoA stays in the cytoplasm. Depending on whether oxygen is present or not, the next step in the catabolism of carbohydrates is the Krebs cycle. The Krebs cycle is also called the citric acid cycle, or tri-carbolic acid cycle. It takes 2 turns for each glucose, and the outputs include 3 NADH, 1 FADH2, 1 ATP, and 2 CO2s. The breakdown of glucose ends here. In summary of the Krebs cycle, this is a part of cellular respiration and the site of entry for catabolism of many non-carbohydrate substances, such as amino acids and fats. It can also be part of anabolic processes, as illustrated to the right. Recapping so far, glucose is being oxidized to CO2 and electrons are being removed. Energy is being harvested. ATP is made directly through substrate-level phosphorylation. High energy electrons are added to electron carriers NAD and FAD to become reduced when they take those electron carriers up. For the process to keep going, the electrons need to be taken up by an acceptor. The energy of those electrons will be captured as ATP in the process of oxidative phosphorylation in the electron transport chain. Oxidative phosphorylation. Here, electrons are offloaded to the electron transport chain. A series of carrier molecules that are in turn oxidized and reduced as electrons are passed down the chain make up the electron transport chain. Energy released can be used to produce ATP by chemiosmosis. A hydrogen ion gradient is created, which flows through the channel of the enzyme that makes ATP. Eukaryotes, inner membrane of the mitochondria, is where oxidative phosphorylation occurs. In prokaryotes, this occurs on the plasma membrane. Here we see the proteins of the electron transport chain. They are referred to as cytochromes, flavoproteins, iron sulfur proteins, and quinones. Electrons are passed on from one to the other. The final electron acceptor of the electron transport chain is oxygen, which is reduced to water in aerobic environments. Nitrate, nitrite, sulfate, carbonate are formed in anaerobic environments, which occurs only in some microbes. This is an illustration of chemiosmosis. Hydrogen ions are pumped across the membrane, and a chemoosmotic gradient of hydrogen ions forms, where you have a greater concentration of hydrogen ions outside the cytoplasmic membrane than inside. Those hydrogen ions will then leak through the CF0-CF1 complex seen at the far right. Those moving charged particles' energy will be harnessed by the enzyme ATP synthase and used to make ATP. This is called chemiosmosis. The summary ATP yield of cellular respiration is given in this table. Take a moment to familiarize yourself with the energy source, carbon flow, and ultimately what is produced in each stage. Fermentation. Fermentation occurs in the absence of oxygen. Fermentation has been used by humans for centuries for food and beverages. Fermentation also has industrial use. Why do fermentation? Cells may be unable to or not have enough final electron acceptors and therefore must do fermentation to recycle them. Cells may be missing components of their electron transport chain. The Krebs cycle enzymes might be missing. And again, fermentation is then required. NADH from glycolysis needs to be oxidized back to NAD to keep glycolysis running. In fermentation, pyruvate is reduced so NADH can be oxidized. This is alcoholic fermentation and is used to make things such as beer, wine, and sake. It's also used for biofuels and consists of two simple reactions. One where pyruvic acid is converted to acetaldehyde, and a second where acetaldehyde is converted to ethanol. Here we see lactic acid fermentation. Lactic acid fermentation produces lactic acid in things like yogurt and cheese. It can also produce sore muscles in multicellular organisms such as you and I. This is a summary of the different fermentation products from your text. Take a moment to review the pathway, the end products, and the examples that follow. Catabolism of lipids and proteins. Large molecules are broken down into smaller molecules outside the bacterial cell. Starch can be broken all the way down to glucose using exozymes. Lipids are broken down to glycerol via glycolysis and fatty acid beta oxidation, acetyl-CoA, and then eventually into the Krebs cycle. Proteins are broken down into amino acids. Amino acids get the amino group and carboxyl group removed, and organic compounds then enter the Krebs cycle. The use of biochemical reactions can also be utilized for microbial identification. Fermentation, amino acid degradation, hydrogen sulfide, and ammonia production are all key determinants for identifying different types of microbes. Photosynthesis. Photosynthesis is an anabolic process. In photosynthesis, organisms use solar energy to produce glucose and oxygen from carbon dioxide and water. Plants and some other bacteria do photosynthesis. Without them, there would be no oxygen to breathe. This is a summary of the reactions of photosynthesis. In the reactions of photosynthesis, carbon dioxide plus water and light energy is used to make glucose, water, and oxygen. There are two parts to photosynthesis, the light-dependent and light-independent reactions. This is a simple summary of the light-dependent and light-independent reactions. On the left, we have the light-dependent reactions that produce electron carriers such as NADPH and ATP, as well as the waste gas oxygen from water. On the right, we have the light-independent reactions, which take in carbon dioxide, ATP, and NADPH from the light-dependent reactions and produce carbohydrates. In eukaryotes, photosynthesis occurs in the chloroplast, specifically utilizing the thylakoids and stroma. In a prokaryote, photosynthesis occurs in the infoldings of the plasma membrane. Light is necessary for photosynthesis, specifically light within the visible light spectrum, as seen here in the colors of the rainbow: red, orange, yellow, green, blue, indigo, and violet. The photosystems, which harness the light energy from the sun or any light-producing structure, such as a light bulb, are bombarded by photons of light, which excite the electrons within them. The excited electrons are released from the photosystems and then picked up by adjacent electron transport chains. The excited electrons go to an electron transport chain and are used to develop a chemoosmotic gradient similar to cellular respiration. If the electrons go back to the chlorophyll, they came from cyclic reactions. If they do not, then they come from non-cyclic reactions and oxygen is produced. The electrons can also go to NADPH to the Calvin-Benson cycle to make sugars. And this brings us to the end of Chapter 8, Microbial Metabolism.